Hello everyone, my name is Mitra KS, member at Shivi Forensics. Today I am going to share some of the MCQs related to the topic Forensic Toxicology. So before starting the MCQ, we will look into the introduction of Forensic Toxicology. Forensic toxicology is the application of analytical chemistry, pharmacology, and clinical toxicology to the purpose of the law. It involves isolating, detecting, identifying, and quantifying drugs, alcohol, poisons, and other chemical substances in biological samples to help determine their role in illness, behavioral changes, or death. Father of toxicology is Paracelsus. He was a Swiss-German physician and alchemist, and he is famous for establishing the core principle that the dose makes the poison. Matthew Orfila is widely recognized as the father of modern forensic toxicology. He was a Spanish-born French toxicologist and chemist, published the foundational Traité des Poisons in 1813, which established the first systematic approach to identifying poisons in the human body and correlating them with legal medicine. So let's start our MCQ. The first question is, which analytical technique is widely considered the gold standard for confirmatory testing in forensic toxicology due to its high specificity and ability to identify unknown compounds? The options are high performance liquid chromatography, gas chromatography mass spectroscopy, UV visible spectroscopy, thin layer chromatography. The correct answer is GCMS, that is gas chromatography coupled with mass spectroscopy is a gold standard because it separates volatile and semi-volatile compounds and provides a unique mass spectrum for each, allowing for unambiguous identification. While HPLC is an excellent separation tool, it is primarily used for quantification and screening and is usually coupled with mass spectroscopy for definitive confirmation. UV visible lacks the specificity needed to distinguish between many complex drugs and poisons. TLC is a simple preliminary screening technique, but it lacks the confirmatory power of mass spectroscopy. So in this slide, I have mentioned the clear definition about all these techniques, a small brief description about all these techniques and why GCMS is considered as a gold standard. And these details are mentioned in this slide. So let's move to the second question. Second question is, what is the primary purpose of presumptive or screening test in forensic toxicology? The options are to quantify the exact concentration of a poison in the blood. Option B, to quickly narrow down the presence or absence of specific drug classes. Option C, to legally establish the time of drug ingestion. Option D, to determine the exact chemical structure of a novel synthetic drug. So the correct answer is to quickly narrow down the presence or absence of specific drug classes. Presumptive tests are rapid, cost-effective methods used to eliminate negatives and identify potential drug classes before proceeding to expensive and time-consuming confirmatory tests. Next question is, what phenomenon describes the movement of drugs and poisons from surrounding tissues and organs into the blood and other bodily fluids after death? The options are postmortem redistribution, bioaccumulation, autolysis, putrefaction. The correct answer is postmortem redistribution. Postmortem redistribution significantly alters drug concentrations in the blood, making interpretation of toxicological results challenging if the samples are taken long after death. So let's look into the other terms mentioned in the options. Bioaccumulation refers to the build-up of substances over time in a living organism and after not changes happening after death. And autolysis is the destruction of cells and tissues by the body's own enzymes and not the movement of drugs. And third one is putrefaction. Putrefaction is the decomposition of body tissues by bacteria which alters the physical state but is not the primary mechanism for drug movement across tissues. So from this explanation of all the options, the correct answer is postmortem redistribution. The next question is, which biological specimen is often considered the specimen of choice for the analysis of volatile poisons such as ethanol, methanol or cyanide? The options are urine, gastric contents, vitreous humor, scalp. The correct answer is vitreous humor, and vitreous humor is a fluid in the eye and it is anatomically isolated and offer resistant to postmortem decomposition, making it highly reliable for analyzing volatiles and drugs. So the other options are urine, gastric contents and scalp hair. The first one is urine is excellent for screening drugs of abuse but is less reliable for determining recent volatile poisoning levels. And the next option is gastric contents. Gastric contents can show what was ingested but do not reflect the active concentration of the poison in systematic circulation. And the fourth option is scalp hair. Hair is used for detecting long-term or chronic drug or poison exposure, not acute volatile poisoning. So here in all these options, the correct answer is vitreous humor. The next one is, what is the standard preservative added to blood samples collected for forensic alcohol analysis to prevent the microbial production or destruction of ethanol? The options are EDTA, sodium fluoride, heparin, potassium oxalate. Here the correct answer is sodium fluoride. Sodium fluoride acts as a preservative and an enolase inhibitor, preventing microorganisms from either creating alcohol or breaking it down in the sample. The other options are EDTA, heparin and potassium oxide. All these three options are an anticoagulant used to preserve blood for DNA analysis. So the correct answer is sodium fluoride. The next question is, long-term exposure to heavy metals like arsenic or mercury is often best detected by analyzing which of the following samples? The options are hair and nails, bile, cerebrospinal fluid, synovial fluid. The correct answer is heavy metals bind to keratin, making hair and nails excellent specimens for determining chronic long-term exposure to these toxins. The correct answer is, here the correct answer is hair and nails. And the other options are bile, cerebrospinal fluid and synovial fluid. So let's have a brief description about all these options. Bile is useful for analyzing certain drugs, but it does not provide a historical record of heavy metal exposure over time. The next one is cerebrospinal fluid. CSF reflects recent exposure and is more invasive to collect. It is not typically used for tracking heavy metal history. And next one is synovial fluid. Synovial fluid is found in joints and is rarely used in routine forensic toxicologic analysis of heavy metals. The next question is, poisoning by carbon monoxide leads to the formation of carboxyhemoglobin, which gives the victim's blood and tissues a characteristic. Option A, dark bluish purple color. Option B, pale grey appearance. Option C, bright cherry red color. Option D, yellowish tint. Here the correct answer is carbon monoxide binds to hemoglobin with a much higher affinity than oxygen, forming carboxyhemoglobin, which has a distinct bright cherry red color. And the other colors, that is dark bluish purple color, is typically associated with hypoxia or asphyxiation where oxygen is lacking. That is example cyanosis, but not specifically carbon monoxide. The other one, pale grey appearance, is not characteristic color associated with carboxyhemoglobin. And yellowish tint is typically associated with jaundice or liver failure, not carbon monoxide. So here the correct answer is bright cherry red color. The next question is, in forensic toxicology laboratories, immunoassays such as ELISA, enzyme linked immunosorbent assay, are most commonly utilized for option A, determining the exact enantiomer of a drug present in a sample, quantifying the precise concentration of trace elements, identifying unknown synthetic cathinones, rapid preliminary screening for drugs of abuse. The correct answer is ELISA is a highly sensitive, high throughput assay used primarily for the initial screening of broad classes of drugs of abuse in biological fluids like urine or blood. The next one is, what term refers to the concentration range of a drug or poison that is typically expected to produce the desired? The options are lethal range, therapeutic range, toxic range, minimum lethal concentration. Here the correct answer is the therapeutic range or therapeutic window is the dosage or blood concentration range that is effective and relatively safe for treatment. The other options are lethal range, toxic range and minimum lethal concentration. The lethal range refers to the concentrations associated with death, not therapeutic or clinical effects. Toxic range includes concentrations that are harmful and lead to adverse effects exceeding the clinical benefit. Minimum lethal concentration is the lowest concentration of a drug that has been reported to cause death, not the intended effect. So the last and final question is, during a postmortem examination, if heart blood is unavailable or highly contaminated, which alternative fluid is frequently collected for comprehensive drug screening and quantification? The options are femoral blood, B. pericardial fluid, C. peritoneal fluid, D. amniotic fluid. The correct answer is femoral blood, that is blood from the leg, is generally considered the preferred site for postmortem toxicological analysis as it is less susceptible to postmortem redistribution than heart blood. And the other mentioned options such as pericardial fluid, while it can be tested and it is not the standard substitute for comprehensive drug screening when heart blood is unavailable. And peritoneal fluid, which is found in the abdominal cavity, is rarely used as a primary substitute for blood in toxicological screening. And the last one is amniotic fluid, and it is relevant only in cases of pregnant descendants and is not a standard substitute for systematic blood analysis. So that's all for this MCQ session. Thank you. Hope this video is useful for your studies.